Fusion, the international science radio show.
Hello, Saji.
Yeah, good morning. I'm CuhaWijay Sathiyajith, a dual tech startup founder, SAAINNOVATE and SAAI.
Saji, you are a researcher who's working on a combined method of therapy and diagnostics for cancer, and you've started your own company to commercialize it. What is Radiotheranostics?
Well, it's a hefty question, but at the same time, I should bring it a complex, a subject into a simplified manner so that people can understand. So if you imagine, before I go into the radiotheranostics, let's separate the word radio and theranostics. So the radio stands for the radio metal, and theranostics is actually a combined terminology for therapeutics and diagnostics in one. So radiotheranostics, it's about a molecular complex that actually you can inject that molecular complex through intravenous injection, and then the molecular complex actually travels through the blood and actually releases the metal when it docks to the tumor. And that radio metal, because that metal is actually emitting radiation, so it can kill the tumor DNA. So it's destroy the tumor DNA. So this is the traditional radiotheranostics I'm talking about. But ours is slightly different. I'll come to that bit later.
All right, so you're looking at ways of treating and diagnosing cancer and many different types of cancer, I understand, with
Yes.
a radioactive molecule that you are specially engineering and you inject it. Is that right?
But I would like to stand correct here because this is highly complex technology, because usual in the sense people call it as this molecular complex as chela, right? I mean the chela, because the chela and the metal comes together to form giving you the complex. Because why this chela? The chela is actually a Greek word for it's actually stands for the chelator. So that means, like, if you imagine like a crab claw, right? So the crab claw is what it's actually mean by. So the crab claw can hold the metal so that it holds in place. So but here, I mean, as I mentioned initially, that the molecular complex that travels to the blood and releases the metal and when it docks to the tumor, and it actually emits the radiation. But imagine that the molecular complex is the chelator itself can act as a drug, not only for one particular type of receptor, but it can target several receptors. So here again, I mean, for the audience who doesn't aware of the receptor concept, let me put it in simple words. It's about, in the tumor, if you take a tumor cell, it will have signaling points, signaling post in the surface as well as inside the cell. So if you can target both these type of signaling post at the same time and simultaneously releasing the metal, then it's actually a dual impact to the patient.
So can I ask you,
yes.
Sorry to interrupt, but I'm just trying to make sure I'm following. So these receptors are part of the cell's signaling mechanisms, and is the cancer using these receptors differently to other cells?
Yes, that's a good question, because when we say like signaling post, or you can imagine like a signaling points, right? So these signaling points are actually overexpressed. We say it overexpressed because it is the number of the signaling points in the tumor cell differs from the normal cell. So these signaling points are actually present in both cells, of course, but in the tumor cell, because of the genetic aberration, is actually overexpressing these signaling points. So
Right. So there's a lot
Yes.
more of them in cancer cells. So
Yes.
more of the radioactive molecule will be taken up into the receptors.
Yes, yes. So the active molecule can take up to the receptors. Why? Because of the molecular complex can actually directly bind to the signaling points. But in traditional manner, what we do is actually we will have a chelator that will have a protruding element where we can have a linker, and then that linker will actually bind to the binding molecule. There will be a targeted binding molecule that actually go and bind to these signaling points. So in other words, it's actually a three-component system. But whereas here, so the three components, that is the chelator, the linker, and the binding molecule. But here, you don't need the binding molecule because the chelator itself is acting as a binding molecule, directly binding to the molecule. So therefore, it actually it's a one component unified chelator. And not only that, it actually the chelator itself can penetrate inside the cell and target the inside signaling points as well.
So
So
I have two questions about this. One
yes,
is that I'm still not quite sure how this is not targeting normal cells with the same receptors. So how is
yep.
it going just for the cancer ones and not for the healthy ones?
That's yeah. So that is how does it happens, because since it's overexpressed on the these particular signaling points are actually highly overexpressed, relatively overexpressed, and so it will not actually going to bind to even if it binds to the signaling points, the normal I mean in the normal cells signaling points, it will not have an impact because it's about the relative receptor density. We call it as receptor density. So the receptor density will be very much higher in the tumor cells. So if we, I mean, this is actually a complex, phenomena. But you can imagine like instead of taking the medicine to the patients, we are trying to bring the patients to the medicine. So it's actually like an inverse thinking that
Mm.
actually brought up this concept. And in fact, I mean, this was a very controversial thinking, because it's actually completely differs from the traditional pathway. So but Professor Rodney Hicks actually invited to Melbourne Theranostics Innovation Center, and where we actually have a in-depth discussion about this. And then when we explained the data and the idea, then it was unanimously accepted. And then on there's several world conferences also have actually unanimously accepted this kind of answer. I mean, in fact, it's interesting to say that it's actually 2023 I actually brought up this sort of concept, but nowadays people are looking at it more into this naturally, but obviously not from a single molecular point of view, but it's actually that they are still using like 3 or 4 components, and or it could be like a decorated molecule. But ours is not like that. It's just a
Mm.
small molecule. That's everything. So
And I'm
that's
just
imaging and therapy in one. So
imaging and
that's what.
therapy in one. Yes.
Yes.
And so, well, I'll ask the second question first. So the imaging
Yeah.
part, what sort of imaging are you doing to see this radioactive molecule?
Yeah, so because usually the radioactive molecules will be emitting different type of radiations like beta emitting, positron emitters and all that. So because of these emissions, we can actually target it through the PET, positron emission tomography aspect. So you can target the PET, and on top of that, this molecular complex need not to be having only the radio metal. It can have a magnetic metal. So therefore, it can act as a MRI contrast agent. So therefore, that's also one thing. And on top of that, this the molecule, the chelator actually itself is acting as a dye, is a visible dye. So because of that, it actually acting as an optical imaging agent. So that means it's PET, MRI, and optical. And then the people who are, SPECT lovers will ask, like, how about SPECT? Of course, it can be utilized for SPECT as well. So that's why it's like multi-modal. I mean, multi-modal in the sense it covers more or less the whole spectrum of thing. And then recently, our recent research is actually expanding to photoacoustic imaging as well. So that is in time to come. We will explore that.
Amazing. Support diffusion by buying from the Diffusion Science Etsy store. Catch people's eyes like flies. I have Hawaiian shirts, t-shirts, pullovers and mugs with many different Venus flytrap designs based on my photographs. Check out the shirts and other Venus flytrap designs at diffusionscience.etsy.com. I'm Ian Woolf. So the less important question is, with my limited knowledge of the chemistry, you talked about chelating. Now, I've heard about chelating agents being used when people have, sort of metal poisonings as an agent that
exactly the concept is the same, similar. Yeah.
Right. So, but I guess my confusion there was you were talking about injecting a metal, but you're also talking about it being a chelator. So can chelators have metals as part of their as part of their structure? Right.
Yeah, not part of the structure. What we do is chelation, because it's actually chelation complex. When you say taking back to the basics on this, like chela is actually stands for crab claws.
Yep.
So that is where I said this crab claws is the chelator that I'm talking about.
Right.
Right. That's because the chelator, the word chelate actually coined from the Greek word chela, right?
Yes.
Which stands for crab claws. Yep. So that the crab claws actually holding this can be actually made to hold the metal because of its different arms. We call it as coordination arms.
Right. I'm with you.
So, for example, as you from your example, like EDTA is actually a commonly utilized agent for depoisoning, like lead poisoning.
Yes.
So the concept is very similar, because we are actually utilizing an EDTA based molecule, and you are actually you can then complex it with PP or lead or or all other metals. I mean, transition to lanthanide elements. I mean, it could be manganese, it could be copper, it could be gallium, it could be actinium, it could be lutetium, it could be 56 Mn, 52 Mn, and 64 copper. We have already proven this, and 68 gallium we already proven, and lutetium 177 recently proven. So it can be combined with any of these metals. And here the 64 copper is actually it's a radiation emitting metal, but it's not like a targeted therapeutic radioisotope, right? So but whereas you can put the 67 copper, which is another isotope of the copper, so that can easily bound with this same chelator, so that you provide the benefit that I told you about, the radiation plus inhibition. So on top of that is a therapeutic part, and on top of that you can image. So in theranostics we say, see what you treat what you see, right? So you can actually see what's happening in the real time. And on top, and most interestingly, this molecule is actually a fluorescence lifetime imaging agent. That means it can be utilized in the live cell imaging. That means how this actually the molecule is binding, that you can visualize it in real time.
Amazing.
So it's not someone's hypothesis or a imagination or anything. You can actually see that. So we are going to produce some images, very interesting images in the short run. So that will be. interesting.
Now,
Hope that
so
answers your question, or
it does
maybe.
answer my question. Yes. Now, I'm trying to recall, so the other thing you've mentioned here, I noticed in the name of the company, you've got the word
Mhm.
AI. Is that deliberate? Are you
Oh,
using AI in
well,
your research?
well, of course, I must stand correct again. The thing is here, like there are two, what do you call, two, I'm actually, as I said, I'm an actually dual tech startup founder. So
Yes.
one is about SAAINNOVATE,
Yes.
and the other company is called SAAI. But
Ah,
as you rightly pointed out, both have the basis of AI, very strong basis of AI, because
yeah.
because this Psi innovate actually stands for self-amplified OGB inhibition. That means OGB is actually a signaling point inside the cell. So which is actually aurora kinase B. It's actually an abbreviated form of the aurora kinase B. So we say self-amplified aurora kinase B inhibition. That's innovation, right? So how do we innovate that? Because I mean, there are people who, I mean, there are already commercial drugs like baracetab is already being utilized to target the inside the cell. But whether the baracetab, how does it targets? Can you image it in real time? No, it's not yet. But probably, I mean, after my interview, people will do that as well. But yeah, but we have some proprietary uh, you know thing, but yeah, coming back to the story, it's about so aura kinase B, so that's one thing. And but in fact, this the whole Psi innovate, how does it originate, is another interesting story itself,
Mm,
because it was at the time where molecular docking and molecular dynamic simulations are not really taken over by the whole scientific community. And these results that comes from the Autodoc Vina or the MyPresto portal and all that sort of it's like similar simulation softwares are not really in tune with that. But so we actually, I mean, initially when I was trying to publish the paper in results in, in RSC Advance. So so when they initially rejected, saying that this molecule cannot be utilized by any specific people, because the thing is like, because it was exciting in the UV, excitation in the UV, and emitting in the uh, in the visible range. So and because of that, so I was actually prompted to add some value addition. So that's how the whole innovation came up. And the innovation from the value addition point of view, because this molecule has the EDTA based molecule, has some very hydrophobic groups which can actually have a non-covalent interactions with the proteins. So we thought like, I mean, maybe we should explain, a bit, explore a bit more on the aurora kinase B, like, I mean, some potent pan-cancer targets, whether
mm,
it can actually target these. So then when we evaluate that, at that time, I don't have any funding. So at the time, so it was like I was actually doing in my computer, and then just when I did the first validation about this aurora kinase B binding, I was not sure, because I thought like, how come a computer can do all this? Like, yes, I mean, initially, I mean, like, yeah, I have got the background, but still, I was not convinced enough. And
mm.
then I actually called Professor Peter Edwards, who is my principal supervisor in my PhD, who is actually a mentor as well, who is also inventor of MyoView. So, so he actually told Sanjit, this is phenomenal, and he asked me from the whole steps I went through, and he was extremely convinced. And but interesting enough, because I actually later on, when I actually found this molecule can actually target the tumor microenvironment as well in terms of fibroblast activation protein. And then I actually went through that. And when I tried to, to see whether it actually does the same thing with the using the same software, and got the validation that it's actually allosteric binding is good enough. But whether that is true or not, I was not. I was skeptic. So then I contacted Professor Ken Herman, who is actually a very strong supporter of SAAINNOVATE. And Professor Ken Herman actually put me on touch with Professor Ube Habakon. So Professor Ube Habakon is actually a pioneer everybody knows in the field. So he's actually a FARP, fibroblast activation protein theranostics. He's quite familiar with that. So he actually asked me to send the whole, the validation protocol, everything, to see whether it actually does that. And we have seen that it's true. So yeah, I mean, but now we will be very shortly, we will be releasing some cell-based assay data. So it's an exciting moment for SAAINNOVATE. We'll see what is the truth. We will know very soon.
Yes. Well, the idea
But
that you're able to get imaging of what's happening in the cell in real time to see what's
yes,
really going on is quite amazing. Is that something that's generally been available before?
yes, it is available. I mean, the technology that I spoke about, like positron emission tomography or magnetic resonance imaging (MRI) or optical imaging (OI) or the fluorescent lifetime imaging (FLIM), all these are available. And
Yeah.
single photon emission tomography SPECT as well. But the beauty lies here. The beauty of this, with respect to our theranostic, lies here because our theranostic agent can actually incorporate all of these modalities. It can work in all these modalities. So in a customizable manner. Sometimes it needs to be customizable radioisotopes to be selected, or sometimes it can be with the one particular radioisotope itself. Even, for example, the 64 copper, which we actually took it as like a diagnostic imaging agent. So it actually worked in hybrid PET MRI. So in Stanford medicine, our validation study has proven that is a 63% regression in 24 hours, which is
So is that your paper from last year?
no, that was the topics in magnetic resonance imaging. That was in the MRI.
Right.
That was actually, so this results has yet to be published because it's currently in a very high profile Nature publication. So it's actually under a long review because of the cell-based assay validation
Yes.
has to be done. So we are actually fundraising for that to complete the job. But because we have animal studies have proven some excellent results, 63% regression in 24 hours in a lung cancer model. And we have actually back validated that data with the PET CT and ex vivo biodistribution and so on. So it was a rigorous data, and it clearly shows it does. But we need to know that how it actually does, whether the mechanism I am proposing is the one, or is there any other mechanism that is acting synergistically to bring this effect?
Wow.
Is the question. So there is partial agreement with the mechanism hypothesis as well, but we need to say that because of the confidentiality, I can't go into detail about what it's all about. But we are quite certain that the truth will be revealed very soon.
Yes. So you've done the molecular assays that are in this paper and also the animal studies.
Animal studies.
Yeah. So
Yeah.
once this paper is published, and I understand that with the review process, you're still doing basically work on the paper to get it up to
Yes.
what they're
Yes.
happy with. Will the next step be human clinical trials?
Yes. Actually, we have, thanks to Professor Rodney Hicks, who actually, after the 2.5 hours in-depth discussion along with the head of clinical operations, so we have decided to start the phase 0, which is the first in human trials. So we are actually capital raising for that. I would really appreciate anyone who is listening could come forward for this fourth generation precision theranostics, so where I can bring the benefit to the patients from the tumor spread and recurrence also to be minimized while you image and monitor them So it will be a very interesting activity and a useful activity for the community. So I hope people can support this endeavor. And in fact, it was more interestingly, it has actually initiated as one particular site in Monash Health, but now there are about 5 sites in Sydney. NSW has joined actually in this multi-site trials. So we have some got some special endorsement from Associate Professor of Professor Kerry Hitos, on this technology. So it is from Faculty of Medicine and Health. So I think we are moving on to an exciting stage.
Yes. So
yes.
you've got the paper coming out in Nature with your cell assay and your animal studies.
Yes.
You're raising money at the moment to be able to do, I know, clinical trials can be very expensive indeed.
Yes.
And there's
About
several rounds.
2.25 million.
Yes, and I was like,
2.25 million US, Australian dollars.
yes,
We
and
have
you've got to
budgeted for
go.
that.
You were talking about clinical phase 0. How many phases do you require for a clinical trial to be able to get the treatment
Good question.
where it could be commercial?
Yeah, great question. So the phase 0, this is actually a de-risk project. We call it as where we can actually find, not that whether the project is going to be successful or not. We know that it will be successful, but to what extent it's going to be successful is the question, right? So that's what we call it as like de-risking the project, so that once that because we also know that there will not be any sort of safety issues, hardly any safety issues, because of the molecule is actually a simple molecule which is naturally available, a molecule that actually produced by combining a naturally available biocompatible elements, right? If you like two segments coming together. So it's so we know that there's hardly any issues, and the toxicology early toxicology studies that we have carried out in Lausanne University Hospital, when we injected about 400 microliter to the mice, it was still survived. So we are quite sure about that. So after this phase 0, then there will be phase 1, and phase 1 and phase 2 can be combined together. So it will be a massive scale. So phase 0, we are trying to recruit about 10 patients, and which is the patient recruitment is not an issue. We have already filed. So our main restriction is the funding restriction from the capital raising side for the clinical trials. That's the only restriction we have. From scientifically, we have all set.
Fantastic.
For that.
So you've got a company producing this new technology. Who's your team?
Yes, so it was actually all started as like a one-man army, as a sole trader business. But then in 2025, I got the whole team assembled, and now we have a very strong business advisor, and who's going to be my director, Mr. Robert Hodges. He's actually a serial entrepreneur. He's actually with us, very strongly involved in this endeavor, and also we also have a support from the angel investment, meaning Strikes Trust. We have actually Miss Olivia Vivian, who is actually a Olympian. So we have a ninja, Australian ninja warrior. Everybody knows her. So she was the first supporter of this technology, this thing, and so we are actually trying to fundraise with this, with the business background, but we have a strong advisory board as well, which comprises Professor Helen Nardal, who is actually the immediate past president of Society of Nuclear Medicine and Molecular Imaging. Everybody knows again her. And we also have the support of Dr. Deepak Behra, who is actually another CRC, clinical research company CEO, and he has decades of experience in this translation from a bench to bedside translations. He's quite involved in that. And we also have strong support from Professor Ken Herman. And also on top of that, we also have Professor Thomas Mead, who has actually gave us the assurance that he would like to support SAAINNOVATE. Professor Thomas Mead is actually a professor of 6 different fields.
Goodness.
So including professor of biochemistry and radiation oncology and so on and all that. So he is very supportive for us to bring that, and he's himself as an entrepreneur as well. So he has about he has sold 6 companies. For 2.2 billion, developing from scratch. So getting his support is a huge blessings for us. And on top of that, we have like 15 global collaborations that I've actually failed to mention, because I will be failing in my duty if I do not mention about Stanford Medicine, because Stanford Medicine actually helped us to do the validation, preclinical validation, as well as proof of concept studies we did with that, and similar to Lausanne University Hospital, which was actually already published in the paper, but in the coming paper it will be Stanford Medicine's contributions will be acknowledged. And also there are UNSW is now helping us, so hopefully we will move forward as it's about science. So I'm waiting to see what the science talk about, because I'm always because as a founder, as a strong founder who believes in integrity more than the intelligence, so we would love to accept whatever the nature going to give it us. But one thing is for sure, that it's all roads points to Rome, as they say. So far, yeah, that's what it is. So we could definitely hope something interesting from the trials.
Wow, that's an awful lot there. So that's a fantastic project. So you've got a sort of a 2-in-1, well, multiple diagnostic, multiple imaging,
All in one. That's what we
all
say.
in
All
one,
in
and
one.
it's the treatment as well. And
Yes.
you've already got a paper on its way with the cell assay and the animal studies, and
Animal studies.
you're
Yep.
almost
Validation
coming up
studies.
to the human clinical trials.
Yes.
And you've got this incredible team of really talented people. It looks like an amazing project.
Yes. Indeed. It's a very exciting project for the whole cancer community. I
Yes.
would say. I mean, not only because we are, and just to before I finish, I should mention also, like, I mean, this is initially we are targeting for cancer, but our success can be expanded to other indications as indicated in the endorsement from our Professor Kerehitos, who
So
actually.
it could be for more than cancer.
Yes, it can be utilized in Alzheimer's and Parkinson's disease,
Goodness.
and also infectious diseases as well. So we have already have initiated the collaborations and everything. Again, the funding restriction did not allow us to explore a bit more on that. But I have actually presented I have delivered talks in SAAIN conference. That is, SAAIN conference is actually targeting neurointerventions
We're about
conference
to get cut off.
in Basel. Yeah, thank you. Yeah.
Sorry. Well, Saji, thank you very much.
Thank you so much for your valuable opportunity and valuable time. Thanks a lot.
Study science, because you will find in the study of science a richer, more rewarding life.